Hello everyone. Today we are going to read about an article from Max Planck Society and the article title is A Beginning Without Errors. This is from the Biology and Medicine section of the Max Planck Society. So this article has been chosen to be published on the yearbook of the 2021 and let's get started. A Beginning Without Errors. It is written by Tommaso Cavazza, Lena Warshaw, Melina Shaw. Sorry for the pronunciation. Um, Only about 1 in 3 fertilizations leads to a birth. Many embryos resulting from the fertilization do not develop further because they have the incorrect number of chromosomes. This is one of the main causes of miscarriages and infertility in many cases. The eggshell had the incorrect number of chromosomes to begin with. However, our latest research has revealed that errors are also often imminent after fertilization. When the genetic material of both parents is combined, this process, which is fundamental to the emergence of new healthy life, is astonishingly inefficient. Once an egg has been fertilized by a sperm, the genes of the father and the mother are combined. Both contribute one copy each of the 23 chromosomes that contain the genetic information encoded by this DNA. The embryo thus inherits a complete set of chromosomes. The precursor cell of an egg cell still contains two copies of each of the 23 chromosomes. In order for eggs to be produced from the cell, half of its 46 chromosomes must be removed. This happens during meiosis, which is a particularly error-prone process. Even in a young woman, 10 to 20% or of the eggs have too few or too many chromosomes. For women over 40, even more than 50%. This is because a woman's immature eggs are produced before she is even born. A 40-year-old woman thus has 40-year-old eggs. The older the woman and her eggs become, the more likely the mature egg will have too many of or too few chromosomes. This is one of the main causes of miscarriages and infertility. We have already discovered several factors that contribute to the high error rates in egg cells. The spindle apparatus, a cellular machine, ensures that a maturing egg contains the right number of chromosomes. This apparatus consists of protein fibers that attach to the chromosomes during meiosis. These fibers pull one copy of each pair of chromosomes to one of the two opposite poles. After that, the cell divides. Surprisingly, Surprisingly, our studies show that the spindle apparatus in human egg cells is quite unstable and often attaches incorrectly to the chromosomes. This leads to errors in chromosome separation. We recently discovered that human egg cells lack a motor protein. Sorry, this makes the spindles unstable. By artificially introducing the motor protein into the egg cells, we were able to stabilize the spindles and reduce errors in chromosome separation. Age-related challenges. Errors also occur because of age-related changes in the chromosomal architecture. They fall apart prematurely even before they are separated. Chromosome separation errors happen not only in the egg but also in the early embryo of mammals. The time immediately after the union of sperm and egg and zygote stage appears to be an extremely critical phase for healthy development. We wanted to find out why this is the case. For our investigations, we analyzed videos of human zygotes recorded by colleagues at the New Edinburgh Hall Clinic in England. Their microscopy videos of human zygotes are routinely recorded for fertility treatments. However, especially for ethical reasons, studies on human zygotes are quite limited. We therefore set out to find a new model organism that could be used to study early embryonic development in greater detail. Together with researchers at the Institute of Farm Animal Genetics at the Friedrich Loeffler Institute, Neustadt, we have developed methods to observe the first divisions of a bovine zygote live. Bovine zygotes are quite similar to those of humans. In that is in example given in terms of chromosome arrangement and spindle organization. The frequency of chromosome separation errors during the first cell divisions is also similar in both organizations. Another advantage of the bovine model system is that we can obtain the egg cells for fertilization from slaughterhouse waste. Lost chromosomes. In the laboratory, we fertilize the bovine egg cells in vitro and used high resolution live cell microscopy in order to follow how the parental genetic material combines. After fertilization, the parental chromosomes are initially present in two separate nuclei. The pronuclei under the microscope, we observed that the parental chromosomes accumulate at the interface of the two pronuclei. However, in some zygotes, individual chromosomes did not accumulate. As a result, these were lost when the genetic material was united. The newly formed cell nuclei thus had too far few chromosomes. As expected, these zygotes then showed developmental disorders. The assembly of the chromosomes at the interface of the two pronuclei thus appears to be an extremely important step. If this accumulation of chromosomes is not successful, errors occurs and embryo does not develop correctly. But why do the chromosomes not accumulate correctly? Our results show that motor proteins and components of the cytoskeleton control the movement of chromosomes within the producer. These are the same elements that ensure that the two producers move towards each other. The unification of the parental genome is thus based on two closely linked processes. However, these often go wrong. Whether an embryo develops correctly depends on a remarkably inefficient process. Our findings could be important for the successful in vitro fertilization of human eggs. For example, we showed that defects in human egg cells can be reduced by introducing the motor protein KIFC1. This could improve the success of fertility treatments. So Here the articles is completed. So next article we will read in next video. Stay tuned to my channel. Thank you.